Thank you. I'd like to start by thanking the organizers for putting together such a fantastic program. I'm really having a blast, and I hope you also, take something away from my talk. So we're going to switch gears a little bit. I'm not going to talk about robotics at all. I'm talking about behavioral variation more generically. So my background is in statistical physics and nonlinear dynamics, but I think as most of you here in this room, I've been fascinated by the structure and variation we see in animal behavior, particularly in natural environments, and how it also reflects a broad range of biological mechanisms that are acting across a broad range of spatiotemporal scales, right? From the motor neurons driving the contraction of muscles, to the coordination of different body parts by spinal circuits, to the modulation of these circuits by neuropeptides that reflect internal states like hunger or stress that sort of structure behavior on longer timescales, and how all of this is also ultimately, provides a substrate for evolution, right? Differences in how animals search for food, avoid predators, and find mates provides a substrate for selection. So there's really a lot to learn by a detailed quantification of animal behavior. But if you're not convinced by that, which I doubt you're not because you're all here, I'll give you a more practical example, which is that of neurodegenerative diseases, which we started to work on a little bit recently. So an example of that is Parkinson's, for which we know that the degeneration of dopaminergic neurons leads to locomotor defects like slowness of movement, tremors, and even complete freezing of gait. And accordingly, treating patients with dopamine agonists can alleviate their treatments. But a major challenge in this field is that the response to the treatments can actually be quite variable and often not work or result in side effects. And I'll argue that part of the reason for this is the fact that the disease mechanisms are acting on brains that are to some respect unique from one individual to the other. And so if we are able to resolve this variation among individuals prior to treatment by, for example, quantifying in detail their locomotor phenotypes, we might be able to better resolve why different patients respond differently to the same treatment. All right, so I hope by now you're convinced that studying behavior is important, but how do we actually do it? Well, as we've seen during the week, in recent years, there's been an explosion in our ability to quantify behavior in detail, again, across a broad range of spatiotemporal scales. This is old news. But a point that I'm trying to make here is the fact that we can really, these days, quantify everything from the minute movements of every single limb to how those limbs build up actions and action sequences on longer timescales to, even as we've heard from Bob Data, almost lifelong, behavioral recording, so spanning milliseconds to life. And this is really exciting because it offers a lot of possibilities, but also a lot of challenges, right? How do we, take this rich, high-dimensional data spanning a broad range of spatiotemporal scales to try to make sense of inter-individual differences in behavior, for example. Now, a common approach to this is to basically collect steady-state statistics. Let's say you do your behavioral experiments, and then you compare the average speed of one individual to the other, or let's say the probability of visiting different behavioral states, as we heard from Bob a few days ago, and to compare one individual to the other. And while these offer very valuable insights on the structure to behavioral variation, they miss out on sort of the dynamic and multiscale nature of behavior, right? So two individuals might arrive at the same average speed, but one fluctuates a lot and the other doesn't. And on the other hand as well, two rats here might have a similar propensity for exploring or searching, but then they might do those behaviors in a slightly different way once we zoom into the finite-scale structure of the behavior. And so if we want to robustly quantify differences among these complex dynamical systems, we need sort of approaches that can resolve their dynamic and multiscale nature from the perspective of the animal as it is interacting with the environment. Now, to make this question a bit more concrete, I'll focus on one specific example to start with of an organism, then we'll move on to others. But we'll start with the larval zebrafish, which was actually the original inspiration for thinking about these questions. And this actually came from the interest of a very talented grad student in the team of Claire Viard in Paris, Gautam, who's now moved on to his postdoc in Japan. And he really wanted to understand basically how the fish's response to cues from the environment also depends on their internal needs. So this kind of interaction between what the animal needs and the cues that it's getting from the environment. And so we collaborated with Mike Orger and João Marques at the Champalimaud in Lisbon, who had collected data from hundreds of larval zebrafish exposed to a variety of sensory contexts. Here I'm showing you just a subset of them, but you had fish in the light or in darkness or with like looming stimuli, hunting for different kinds of prey. There were 15 or 16 different sensory contexts and a total of about 500 fish. And for each of these fish, João had used basically an online tracking algorithm that detects these bouts and measures the curvature of the tail along these 8 points as the fish move. So as you can see, they move through these kind of discrete bursts of tail motion. Here's an example of a sequence of bouts that the fish does. Here I'm just plotting one of the tail angles, but he measured 8 of them. And this is sort of the corresponding trajectory that corresponds to this sequence of bouts. And here there's kind of just a zoomed in version for you to see the details of the different kinds of bouts that the fish is doing. Yes?
Why tail angle rather than curvature, which is more natural and invariant?
Good question. I think, you know, they started by measuring the tail angle. That's the data that... Relative to, so from one element to the other. So I guess it's kind of like curvature.
It's a discrete version.
Yes, exactly.
But that's a spatial function. So where are they measured?
Yeah, so it's a spatial function that goes, basically they're measuring the change in angle. So they're measuring, you know, these two vectors and then the angle in between them.
I understand.
As a function of where you are along the tail. Yeah. But I'm looking at an animal-centric perspective. So I'm putting, you know, I'm not looking from the lab space.
Maybe I'm missing something, but tail angle is a function of space and time. And
yes.
It's only been plotted as a function of time.
Oh, yeah, sorry. So there's several, yeah, that's what I was saying. I'm plotting just one angle here,
thank you. But
there's 8 for each of these bouts. Yeah, so you have this 8 cross 175 is the number of frames that they took as capturing the average duration of a bout at this sampling rate. Yes, sorry, I didn't get your question. All right, so we were handed these, you know, discrete time series of these high-dimensional bouts. We have 500 of them. Here's 2 examples. And basically, we wanted to understand, you know, how can we measure the differences between individuals? Do we look at the fine-scale details of every single bout and the different types of bouts that they use? Do we look at sequences? Do we look at how the entire thing gives rise to different kinds of exploration strategies on long time scales? Well, ideally, we would like to work with a representation that allows us to sort of span through the multiple timescales of behavior so that we don't have to make an a priori decision about what matters for the animal. And so we devised an approach that is rooted in statistical physics to try to sort of resolve the multiple timescales of the behavior. So I'll first describe this approach in an abstract sense and using a time model, and then I'll come back to the fish in a second. All right, so I'll be thinking about these bout observations as some function of an underlying state that I don't have direct access to, but I'm thinking about this state as evolving according to first-order dynamics, meaning that I'm having some kind of, you know, Platonic view that if I measure everything that is to know about the fish, then I can make a prediction about what it will do next. So I don't know what the state is. I don't know what this functional form is. I don't know what these parameters are. But the idea is that if we were able to encode the recordings into a dynamical system, that then we can compare the dynamical systems to one another and encode sort of the multiple timescales of the behavior of each individual. And so in essence, what we're trying to do in a more abstract sense is to solve this inverse problem where you can imagine that you have a set of observations, time series observations coming from an unknown parameter space that is varying. And I want to do something to this time series so that I can reconstruct the fact that there were these underlying parametric control differing from one time series to the other. Yes?
Is F assumed to be deterministic?
No. No, F could be an Unraveling equation. No, it's a very generic formulation. Yeah. I'll get back to that in a minute, actually. All right, so, you know, how would you do this? How do we compare, even if we were able to measure these or fit these models, how do we compare the dynamics? Well, there are several challenges, right? So the first kind of obvious one is the fact that measuring distances in parameter space can be misleading, right? So if you're close or far away from a bifurcation point, that means very different things for the dynamics, which I'm illustrating here by classic logistic map transition to chaos, where you can see that the fixed point structure, if I change by the same amount this parameter R here or here, it has a very different impact on the dynamics. So we need to be careful about that. We also have finite samples, so we need to have some way of measuring whether two observations could have been generated by the same underlying stochastic process. And finally, we also want to account for the multiple timescales of the behavior. All right, so how do we do this? So to try to sort of capture these three things, I took inspiration from some computational approaches in molecular dynamics and worked with a equivalent representation of the dynamics in terms of this transfer operator that involves probability distribution functions. So instead of thinking about the nonlinear evolution of the state space, I'm going to think about the linear evolution of the space of probability distribution functions. In a way that basically, this linear operator is encoding those nonlinear dynamics. I'll get back to this in a minute. But if you can do this, then you can, on one hand, sort of make faithful predictions by sampling from the evolved probability distributions. But importantly, for our task of thinking about the multiple scales, we can directly use the diagonalization of these matrices or operators to directly sort of coarse grain our dynamics. By construction, the eigenvalues of these matrices correspond to timescales of the system. And so we can directly extract global nonlinear modes of the dynamics that are evolving on different timescales so that we can hopefully resolve long-lived sequences of behaviors from faster timescale fluctuations. All right, so I'll jump into a time model for intuition and then come back to the fish. So, yes?
Just one place. What assumptions do you need about the underlying dynamics to have that this evolution operator is linear?
None.
It's fully
I'll give you an example. Yeah. So let's say that you have some random Langevin dynamics overdamped on a landscape that looks like that. I designed it like this to capture some of the properties we think might exist in behavior. So you can think about these different wells as different behavioral states. Of course, this is a very crude approximation. But then basically, the system is trying to go down this gradient, but it's getting kicked by noise. And so the barrier heights and the noise set the transition timescales between the different behaviors. So the system will spend some time here, then it will hop there, then it will hop there, and so on and so forth. And there's also a bit of a hierarchy of timescales embedded into this landscape in the sense that there's a higher barrier that it's harder to cross, so induces longer timescales, and there's a shallower barrier that is easier to cross, and so there's a faster timescale. And so that operator that I was telling you before, you can see directly, and the reason why I'm working with this is because we know what the density dynamics look like, which is just the Fokker-Planck equation. And so if you can basically do a finite difference approximation of the left-hand side here, this is basically your infinitesimal generator, which if you put in discrete time is your operator.
You need something which is local.
Local, I guess. Yeah, in the sense that you mean because of the derivatives. Yeah. Trying to think, maybe you can give me examples of non-local.
Where you have memory.
So where you have memory, then you could expand your state to include that memory. So I'm working already under the assumption that I have like all the degrees of freedom.
Guess what I just add to what Francesco was asking,
Yeah.
is replace the potential gradient by a kernel, so convolution in time, because the system does what it's doing now as a consequence of what it did in the past. Never make it non-local in time.
In time, yeah. But this is also a matter of representation, right? Because I imagine that you might be able to, include that as an extra degree of freedom. And I'm working under the assumption that I have all of them. Yeah. And so then you have just a first order Markovian dynamics.
Increase the state space.
Yeah, I'll get back to that in a very concrete example, actually. But that's a very good point. But so, this is our transfer operator. Turns out that you can estimate this from data efficiently. The simplest way to do it is to just discretize your space and essentially count the transitions among different state space regions. So you need some data. But then once you have that, then you can look at its eigenspectrum. In this case, we know more or less what to expect. There are two long time scales, one that corresponds to the longer barrier and another one that corresponds to the faster barrier. And then a whole continuum of harmonic relaxation modes for each of the wells. And the corresponding eigenvectors then capture different properties of the dynamics. So if I look at the right eigenvectors, for example, here I'm just plotting the trivial one, which is a steady state distribution. So this matrix always has an eigenvalue 1, which corresponds to the eigenfunction that doesn't change when I act on it with this operator. So that's the steady state distribution. In this case, we know that it is given by the Boltzmann distribution. So here I'm just comparing Boltzmann distribution with the eigenvector. And on the right-hand side, I'm plotting the left eigenvectors, where you can see that basically combining the first two non-trivial eigenvectors, we can effectively define, 3 different regions of this space that correspond to the 3 different wells, basically depending on, If this is close to each other, then we're on this well. If this is high and this is small, we're on this well. If both of them are high, we're on this well. And so we can use the structure to the eigenvectors, using some fancy clustering approaches to identify the metastable states of your system. And you can play this game across multiple timescales. So in practice, what we do is that we effectively build a fine scale discretization of our operator. Then, for example, we can use the longest lived eigenvector, which is this blue line here, to identify the first energy barrier and get a two-state Markov chain that essentially captures the transitions between this part of the domain and this part of the domain. Subsequently, we can also take information about the second non-trivial eigenvector to subsequently split the right-hand side of the domain into these two wells. And you can play this game. In this case, there's not much more structure happening, but you could play this game ad infinitum with an increasing number of coarse-grained states capturing faster and faster timescales. And so in the end, we're going to be working with this coarse-graining operator that takes the structure of the eigenvectors to build an effective reduced order description of our system, leveraging this notion of timescale separation to build effective models that capture the dynamics on different timescales. All right, let me show you how this can become useful when we're then comparing different time series. So I cooked up a simple example where it's the same dynamics, but now I'm modulating the minima of the potential. So there's 3 different minima. These 4 different locations correspond to different parameter regimes. You can think of this as like different behavioral strategies that the animals have to do. The blue and the orange one share the first well, but differ on the second well. The same is true for red and green. So there's some structure to this variation. And what we're going to do is basically take time series observations sampled from this Langevin dynamics and try to see whether we can say anything about the underlying structure to the dynamics and the variation. And so the way in which we decided to do this in a way that explicitly captures the multiple timescales is to effectively build this hierarchy of coarse-grained Markov models with an, again, increasing number of states capturing faster and faster timescales. And then for a given number of states, let's say for Q equals 2, then just measure the total variation distance between the Markov chain. So this factor 1/2 just makes the distance be between 0 and 1. And so if we run this calculation for a 2-state description of this system, where these are the 2 states, then we can basically see that we can distinguish blue and orange from red and green. So there's high distance here, low distance here. But we can't really resolve differences between blue and orange because those differences are lying on a finer scale. So they're lying on the subdivision on the right-hand side domain. Yes?
Can you remind me, what are the indices A and B?
A and B are multiple simulations of the system with different parameters. So it's just, it's the indexing of how many simulations you have. Yeah. Thanks for the question. And when we go now to a finer scale resolution of 3 states, we start resolving also differences within this blue and orange regime. And you can clearly see that by the block diagonal structure here. And that's coming from the fact that we are resolving the finer scale dynamics that we didn't have before. And so in this way, we're also identifying the timescales in which different structures are emerging in the variation among dynamical systems. All right, so we have this way of measuring, distance between Markov chains at a particular coarse-graining scale. But, we also have this thing that varies as a function of coarse-graining scale. So how do we deal with that? So one way to deal with that is to do the following. So basically, for a given coarse-graining scale, you can imagine this as defining some abstract space where the location of each point corresponds to a particular time series, and the distance between them corresponds to how far they were at that coarse-graining scale. But because we're working from finite time series, actually the exact position of every point in this space has some uncertainty to it because we don't have an infinitely precise measurement of the parameters of this Markov chain. And so we try to effectively account for that by basically designing a kind of like a null hypothesis testing approach through bootstrapping that tries to quantify whether 2 symbolic sequences could have been generated by the same underlying dynamical model. And so this gives us this kind of effective length scale epsilon below which I can't really resolve differences in the dynamics. And then we can run this calculation as a function of coarse-graining scale. This is what I'm showing you here on the right. So in blue is the distances measured from the underlying dynamics. In epsilon is this kind of effective noise scale of uncertainty. And as you can see, basically for low enough coarse-graining scale, so when we're working with 2 or 3 states and so on, there's a big difference between the blue and the orange curve. So we can resolve differences in the dynamics. Notice also this increase from 2 to 3 and then a plateau, which is reflective also of the structure to the dynamics. But we can also show, and you can intuitively understand that for when Q becomes very high, when Q goes to infinity, we have too many states to resolve from the data. And so eventually these two curves must collapse because the distances become, indistinguishable from noise within the finite observations that we have. And so you could imagine basically integrating the area between these two curves as a scalar sort of measure of the dissimilarity between the time series. And so basically that's what it's going to look like, where we're basically penalizing coarse grading scales that cannot be very well resolved from our finite data. And so in this way, we basically can, construct some kind of, multiscale dissimilarity, let's say, between different time series observations. This looks very similar from, yes?
An intuition for, I understand the right-hand side, but I'm not getting a good sense of what these distributions on the left-hand side are, where the black dots are eigenvalues. What are your axes?
Let's see. So you can think about, you've measured all your distances. You have a distance matrix, and this is kind of embedding that distance matrix into a 2D space.
Between pairs of samples from two runs from the same distribution. Generating
yes. So, I mean, every point corresponds to one sample, and by taking the distance to every other sample, you can embed that into a metric space. But this is just an abstract representation. It's not a direct embedding. I'll get to the direct embedding later on. Any other questions here? Please feel free to interrupt because it's the first time I'm presenting this. I'm not sure if it's clear. Yes.
Makes sense why epsilon is increasing as you increase the number of coarse grain states, but I'm not so clear on why the distances would necessarily increase.
Yeah, so basically the more states you have, the more ways you have to be different. And so the only reason here why this doesn't increase is because the structure of the landscape has these 3 metastable wells. So there isn't anything more to learn about sort of, or very little to learn about, a finer discretization of that. But once you get to the noise limit, then you start scaling with the number of states because it's completely unresolved. So up here, basically you just start growing linearly. Yeah. All right, so we can combine this into this multiscale metric, and then, going back to your question, we can use different embedding approaches to sort of map that onto, the principal axis of variation. So we're using the diffusion map algorithm. So the idea, you can think of this as like nonlinear generalization of multidimensional scaling, or PCA. Where basically what we do is that we take those distances and then we basically do a random walk on this curved manifold and try to find the major axis or the slow modes of the diffusion process itself on the manifold. And so what I'm showing here is something kind of analogous to, a variance captured by different modes, where you can see that there's basically 2 main modes that survive and then everything else is noise. And when I plot the corresponding eigenvectors, then I have these, 4 clusters appear, which is pretty obvious from there. Also, the fact that the first mode is much larger than the second one, which you can also get from the eigenvalues. And this reflects the fact that this first mode is capturing differences along this first well, which is the major difference in the dynamics, whereas the second one is capturing mostly differences that are happening at the finer scales. Also notice the fact that even though we had 3 minima that we were modulating, they were living on a plane, the modulation. And that's also why, in the end, we have a 2D space of variation among the dynamics. All right. Any questions at this point? I'm going to show you now in the fish. Yes?
I don't think I fully understood the 3 minima versus the 4 clusters you're seeing here.
So there are 4 different parameter regimes that I'm modulating. In principle, I'm modulating each of these things, but I'm not modulating them independently in the sense that 2 of them are a function of one another. So they're living on a plane. Yeah, that's it. It's not super relevant for the fish stuff, though. So let me move on to the fish. So going back to the fish, we had these, sequences of behavior, and going back to your question, Madhavan, about memory. Here we don't have access to the underlying state space. We only have access to this measurement function of something that we cannot measure. And so instead of trying to naively just build a Markov model from one bout to the next, which is not going to do a very good job, we try to incorporate also basically further temporal information that sort of encodes the effect of these unobserved degrees of freedom onto the dynamics of the ones that I see. And so this is basically, an idea that has been in the dynamical systems literature for a while, of time-delayed embedding, where the intuition is that basically I'm going to construct a higher dimensional space that includes also time delays of whatever I'm measuring. And by doing this, basically the intuition is that if I just measure one single bout, then there's a lot of uncertainty about what the fish will do next. But it turns out that if I include, past history into an expanded representation, this can help me narrow down my prediction of the future. And we can measure that directly by computing the predictive information that is sitting in past bouts. So basically, it's a measure of how much you reduce the entropy over the future based on the fact that you know the past, as a function of how many time delays you take. And you can clearly see that if I don't take any time delays, I'm not very predictive. And if I go up to 4 or 5, then I'm increasing my predictability over the future and getting Markov models that are, maximally predictive in this sense. And so this defines a new representation for my behavioral state, which is now sequences of 5 bouts. Then I can collect from, yes?
What physically that time delay correspond to in systems?
Yeah, unknowingly here, we didn't have the, physical units of time. We're working in bouts space because this is the data that was given to us. It's like a discrete map type approach. But to give you a rough idea, it's maybe on average about 4 to 5 seconds.
Much larger than an individual C start.
Yeah, so the C start will be 100 milliseconds or so. So it's already, clearly there's memory that extends beyond that, the C start.
Do you have a sense as to what that 5 seconds comes from? Where that comes from?
Not really, no. I think we would have to do some perturbations to see. But clearly the animal is not just making the decision to do a C start, but it's planning whatever it's going to do after on a time scale of roughly 5 seconds, I guess. Which yields some predictive power on this timescale that you don't have if you just look at the individual bout level. Yeah, but yeah, biologically, I'm not really sure exactly where the timescale is coming from. That's why we should look at the brain. Yes.
This is actually emerging from the geometric constraints of the arena in which the experiments are being performed. Is it a small well or what?
Great question. So there's a range of arenas here. Some of them are circular, some of them are square, some of them are very narrow, kind of, direct 100-meter race scenario. So there's many different arenas. We've tried the analysis on the different conditions, and always you get more or less the same time scale. So it doesn't seem to matter too much on that. It's quite stable, this value, actually. And we also worked with data from other people, including data from our lab. It's going to be always between 5 and 6, 7 seconds, something like that. Yeah, yes?
Why is it decreasing?
Yeah, so unfortunately, eventually you start hitting finite size effects because you're expanding the dimensionality of your space. You need more data to be able to infer those entropies properly. And so this is as much as we can resolve. Yeah. Ideally, if you have infinite data, if we run this on a time model, and if it's a deterministic system, then it just plateaus. But yeah, I think in reality, when working with finite data, eventually you start running into finite size effects in the estimation of entropies. Yes?
Is there an effect of noise that is being captured in the shape of that curve?
Probably. It's hard to be super definitive here. So there's some work from the 70s from Pierre Gaspard that looks at basically the behavior of estimators of entropy as a function of spatiotemporal scales for different classes of dynamical systems. And you have different scaling laws depending on the class of dynamical systems that you're looking at. But it's very hard to be super definitive here with the finite amount of data that we have. Yeah.
Are you picking the maximum then? Or are you picking like the first?
I'm taking the maximum, the absolute maximum here, I think. Or maybe, I think I'm taking 5, but, there's a very not big change. And it doesn't change anything for what I'm going to subsequently do. You can pick anywhere from 4 to 7, and it's basically going to be the same thing afterwards. If you pick one, then you run into issues. Like you try to build a Markov model and it's not going to predict very well. Like it predicts on very short time scales, maybe the time scale of 5 seconds, but not beyond that. Yeah. All right. Yes.
Does the error bar include the variation between different joint angles?
Variation between different joint angles.
I don't know if you...
So actually, no. So we're working with, it's a very high dimensional space of bouts. So it's a spatiotemporal object already. And so we're working already with a low dimensional representation of that space and then sort of building sequences of that. Yeah. So I'm not sure if these error bars include that noise. Basically here we're computing these error bars by estimating this across every individual or every condition, I think, and then sort of bootstrapping across conditions. It's across, so the representation is 8 joint angles cross 175 frames. So it's the entire body. Yeah. I mean, up to pectoral fins and eyes, we're not taking that into account. Yeah.
Can you say something about the assumptions that are needed for this to hold? Like, do you assume stationarity of behavior over
Totally.
time?
Yeah,
Does it
it's-
support like switching between two different behavioral states?
Switching you can have as long as that switching is happening on a mixing time that is faster than your observation time. So I'm assuming some stationarity. I mean, and that comes directly from, where is it? Maybe I have to go way too much back to get there. From here, right? I mean, this class of dynamics in general here, this is always going to have an eigenvalue 1, which is the steady state distribution. If you want to make this, non-stationary, then you have to make this operator time dependent. That's something that we've worked on. I don't have any slides on that here, but in principle, you can allow your transition operator to become time dependent, which is hard to infer, but you can constrain it. But I'm not going to worry about that right now. I'm just going to see whatever can I capture with my stationary description, and then what are the differences between individuals. But yeah, that's a great question.
I think my animal's like adapting to its environment or has circadian rhythms or I guess circadian.
If you see that adaptation more than once, then it's fine. It's really about whether these dynamics are mixing or not. In the sense that if there's a circadian cycle, but I'm looking at this animal for 10 days, then I'm seeing this multiple times, so that's okay. But if I have the circadian cycle, but I'm looking at the animal for 6 hours, then this is just going to be a non-stationary transient that I cannot capture with this. Yeah, it's an excellent point. It was subsequent work that we've done afterwards to try to also capture those timescales, but I'm not going to talk about that here. All right, excellent. All really good questions. So it's a sign that you're following. All right, so then we collect these 5-bout sort of representation of the state. We count these transitions, estimate this transition matrix, and then look at its eigenvalues and eigenvectors. And the cool thing is that once we do the calculation, we find that even though, as you mentioned, most of the research in zebrafish larvae focuses on single bout responses to stimuli and stuff like that, they actually seem to engage in persistent long-lived sequences of bouts. So when we plot the projection along the longest lived mode of the dynamics, we see minutes-long transitions between sequences where the fish is mostly meandering with this more turning behavior and local search, and sequences where the fish is mostly smoothly cruising. And so this is what we find on the longest of timescales. There's also other interpretable modes that we can extract from this analysis. For example, if we look at the second mode, so here I'm plotting the first, second mode and color coding it by speed, you can see that there's also some persistence over time for either swimming fast or swimming slowly, as well as, for example, persistence for moving left or right, which is encoded in the third mode. So there's this hierarchy of timescales, longest one being this kind of local versus global movements, or with more reorientation events or not. Second, slightly faster timescale, modulation of speed. Third, slightly faster timescale, modulation of heading direction, and so on and so forth. And so by doing this kind of calculation, we can effectively build a continuous low-dimensional representation of the long timescale properties of the dynamics of these fish, where what I mean by that is the fact that if I know the values of phi1, phi2, and phi3 at any given point in time, I can tell you what the fish is doing while keeping all the precise information about the fine scale details of the bout. So I didn't impose the coarse graining. This came from the structure in the dynamics. All right, and so as I told you before, we can then use these eigenvectors to try to build coarse-grained models of the behavior on different timescales. So for example, if we forget about all the fine scale details and just put everything into 2 boxes of cruising versus wandering, and so basically then we can infer these 2-state Markov model for every individual fish and see how well can we predict the dynamics on these timescales. So what I'm plotting here on the right-hand side is basically the mean sequence length, so the number of bouts on average that the fish spends in a particular state. Each fish has 2 points, one for cruising and the other one for wandering. And on the y-axis, we have the result of simulations, where you can see that we're accurately encoding the dynamics of each individual across a broad range of timescales, from a few bouts to a few hundreds of bouts. Any questions here? All right, so we can do the same then, across different timescales. This is just kind of an illustrative picture where basically the idea is that we can go to finer and finer coarse graining scales, capturing faster and faster timescale information. And then this is really cool from the perspective of studying variation, because we can look for every pairwise comparison, where is this variation emerging, at what time scale, which is also important for thinking about the underlying neural processes that might be varying from one individual to another. And so I ran the same calculation that I just showed you in that toy model, so measuring this kind of multiscale distance between fish. This is the distance matrix across all the fish. Again, so each entry here is the pairwise distance between fish across all these timescales within the experimental timescale. So I'm not looking at non-stationary properties, it's only the stationary properties of the dynamics. You can see there's a lot of structure. We can make sense of that structure again by doing the same calculation as before. So looking at the eigenvalues basically of the diffusion map, where we can see that this variation is structured. It's not like it's lying uniformly in all modes. There's a few modes that capture most of this variation, telling us that this variability is structured. So there are certain modes of variation that are more important than others. And so instead of just treating this behavioral variability as noise to be averaged away, we're trying to, we're trying to uncover the structure to this variation and how it might reflect underlying control mechanisms. All right. So as a first attempt to try to sort of understand what this structure might be, we can look, for example, at the first two modes. So again, each point here is a fish. If they're close by, their behavior is similar. If they're far away, their behavior is very different across the entire experiment. You can roughly see that there are regions in this space that are more densely populated than others. We can try to see what the fish is doing in these different regions. These are simulations from the fish in different parts of this space, where the fish that are here on the top right side, they mostly do local searches. So they do a lot of reorientations and slow movements and stay like densely search space. And then you have two separate modes of more global dispersal. One that has a lot more cruising, so the fish have more smoother, persistent motion, and another one that has a lot more sort of faster meandering trajectories. And so to try to understand why the fish would use different strategies, we can look at how they're being used in these different sensory contexts, right? So this plot is a bit difficult to digest, but I'll try to navigate you through the most important structures here. So each of these tick marks represents the average behavior of the fish that were in a particular setting. One thing that is important to keep in mind here is that at this developmental stage, the fish still have a yolk sac, so they can feed themselves. They don't have to actively hunt. But in some of these conditions, fish have seen prey, and in others, they haven't seen prey. And this seems to be one of the major determinants. So all these conditions here on the right, the fish is either sort of hunting for paramecia or rotifers, or they have seen rotifers in their past developmental history, but during the experiment itself, they're just freely exploring in the light. And so what's remarkable is that they're basically in the same condition as the fish that are here on the left, which are also exploring in the light. The difference between them is that some were raised with live prey, others weren't. And this really changes dramatically the way in which they explore space. Might also have impacts on their developmental trajectory. We couldn't really dig into that yet, but so the bottom line, more or less, conclusion from this plot is that there's a major difference between sensory environments where the fish have never been exposed to prey, whether there's prey in the arena, or they were raised with prey, but there's now no prey in the arena. There's also then some more subtle differences between aversive stimuli versus appetitive stimuli and stuff like that I wouldn't get into. But so clearly, there's an effect of prey and also possibly developmental effects driving the variation among individual animals. We can see that more clearly as well if we focus on, for example, one of the conditions where the fish is hunting for prey. So all of these fish that I'm plotting in both sides, so this darker marks is a particular fish. There's a distribution. All of these are in the same environment, so they're all with roughly the same distribution of paramecia around them. The difference between them is that these guys were not raised with prey, and then these guys were raised with prey. And clearly there's sort of a, convergence of their behavioral phenotypes that is driven by the fact that they had this past experience of this prey, which pushes them into the part of the phenotypic space that we expect them to be if they want to hunt in a dense distribution of food, which is to do more of these local searches. Yes?
When you say past experience, how long ago was that experience? Was it 5 minutes earlier?
It was from 4 days on.
Wow.
They started being fed live prey.
But between the experience of live prey and this experiment?
A day.
A day. So
Yeah.
They maintained that experience.
Yes, exactly. So basically what happens between these two is that the feeding protocol was changed. So in one of them, they didn't fed them, and in the other ones in their home tanks, they just put some paramecia. And so that happens at day 4 and 5, and then the experiment is done at day 6. Of course, we don't have full control over the entire experimental apparatus, but as far as we can tell, this is the major difference between the two conditions, is that in these guys from 4 days on, they started to be given live prey, and these guys weren't. And so I find 2 really interesting things about these plots. One of them, of course, is this kind of convergence of the variation that you see from the past experience. But also the fact that, even here, not all the fish are doing the thing that we expect them to do in this environment, right? A lot of them are in parts of this space that are more inducive to like dispersal-like behaviors. And so we were kind of puzzled by this. But the cool thing is that the fish have a tail when they're trying to hunt. They converge their eyes on the prey. And so we can look at their eyes to get a sense for how much they're actually even trying to hunt or not. And when we do that and overlay that on the same plot, we find that the fish that are here in the top right corner are the ones that are actively trying to hunt. And we could tell that basically just by looking at their behavior. Whereas the fish that are more on the bottom side of this plane, they're also not actively trying to hunt. So they're possibly in a different kind of internal state, either they're stressed or they want to explore space or whatever, that pushes them into a part of this phenotypic space where they're also not actively trying to hunt. And so clearly there's evidence here for internal states that are overriding these external environmental inputs and giving rise to variation among individuals. Now, unfortunately, at this time with this data, we couldn't access directly the underlying mechanisms that are changing between these individuals. I'm going to be very brief here, but this is something that we're now exploring in collaboration with Drew Robson and Jennifer Lee, who can measure simultaneously neural activity in the whole brain while the fish is freely moving. And so this is the work of Leo, who's been starting to sort of find some interesting differences in the activity of certain energy populations in the dorsal raphe and how that maps onto different fine scale structures in the behavior and distinctions between more quiescent-like states and active hunting-like states. But I won't dwell too much into this. I wanted to give you a completely different example of where doing this kind of analysis has given us a deeper mechanistic understanding of the underlying control variables. And so to do that, I'll switch completely from a vertebrate to a unicellular organism. So I know this conference was supposed to be about animal behavior, but I hope you allow me to talk about bacteria. So that's the sort of the E. coli. And the cool thing about the E. coli is that Thierry Monet at Yale has done simultaneous measurements of the motility of these bacteria, but also the gene expression of key chemotaxis proteins. So E. coli has been used for a long time as an interesting model organism to study chemotaxis. And they do that by essentially sort of modulating the rate at which they tumble. So as you can see, basically they have these multiflagella that can rotate in different orientations, and they can bundle these flagella to run mostly forward, or they can unbundle these flagella to essentially tumble in space and choose at random, at least that's the description, a different direction. And by essentially modulating how often they tumble, they can navigate up a chemical gradient. So if they're going in the wrong direction, they'll tumble more. If they're going in the correct direction, they'll tumble less. And in this way, they are able to find in the cool side of the chemical landscape. But so we decided to work in this system because, unlike the vertebrate with billions of neurons, here we understand the control machinery of this chemotaxis network much better. There's order 10 chemotaxis proteins that are working in this chemical brain and resolving this task of going up a gradient. And so Thierry had engineered these bacteria to overexpress either Key-B or Key-R proteins. So these are two proteins that basically act on the adaptation of the bacteria. So basically how it works is that Key-R is constantly demethylating membrane receptors and making them more sensitive. So this will increase the tumbling rate. Whereas Key-B, when there is a tumble, it feeds back onto the membrane receptors to methylate them and make them less sensitive. And so this reduces the tumbling rate. And you can kind of see that here by looking at how the expression of these proteins maps onto the cell behavior. So when Key-R's expression is larger than Key-B's expression, then you have this broad distribution of tumble biases. So tumble bias here is roughly like tumbles per unit time. So the bacteria is tumbling more when Key-R's activity is high. And when Key-B's activity is much higher than Key-R, then there's a reduction in tumbling bias because basically you're reducing the sensitivity of the membrane receptors and you're going to tumble less. And it's by basically using this machinery that the bacteria can actually adapt to different chemical gradients very efficiently. All right, but for us, this was cool because, Thierry had measured the motion and had measured directly this sort of single cell fluorescence of the expression of these two chemotaxis proteins. And so what we tried to do is to basically, rerun exact same analysis that I've just showed you in the fish, except that instead of working with bouts, now we're working with this egocentric velocity vectors. But we play the same game where we build this time-delayed embedding and Markov modeling to look at the long time scale properties of the dynamics. And what we find is that basically there's the slowest mode effectively recovers this run-on tumbling canonical description of the bacteria. And then there's other finer scale modes that we find interesting, and we're now trying to understand with Thierry how this could be modulated by how many sort of flagella are unbundled. Well, clearly there's some persistence for also an average speed state, and also for either more directed runs or more curved runs in different directions. So here, I'm basically just plotting the heading change against the speed and looking at the different eigenvectors. And so, again, we can do the same calculation as before, infer these hierarchies of Markov models from each trajectory of each individual bacteria, and then look at their differences. So this is, again, the distance between individual bacteria. And we find the low-dimensional variation space, with one mode being much more important than a second mode. Here I'm just plotting them against each other and color coding them by the diffusion constant so that you get more or less an idea of the different kinds of phenotypes. So basically, here the bacteria is tumbling a lot more, here it's tumbling a lot less. But then the cool thing with this data is that we can also then subsequently look at the gene expression and overlay it here. We basically uncover the fact that the major mode of variation is determined by the expression of key B, whereas the second mode of variation has some relationship with key R, but only when key B is large enough. And this is something that it's hard to see directly from the biochemical network, but once we looked at it in more detail, we understood what's happening. It's the fact that basically key R acts irrespective of what the bacteria is doing. So it's constantly just making the membrane receptors more active. And so if there's no key B to counteract that, then basically the bacteria is just pushed into this steady state limit where it's just tumbling all the time. And that's also visible here by basically the compression of the variability on the space where key B is small. And you can vary key R as much as you want. That's not going to do anything to the bacteria if you don't have key B to counteract that. Whereas when key B is large enough, then you put the system in the right dynamic range, in which then variation in key R starts becoming important and leads to differences in how the bacteria are behaving. And so clearly, there is a difference between these two chemotaxis proteins and how they affect motility. And we're getting this directly just by doing this unbiased analysis of their behavior. All right. So before I conclude, I'm not sure how much time I have.
5 more minutes.
5 minutes. Okay. Perfect.
Questions.
Yeah, that's fine. Yeah, so I'll go back all the way to my introductory slides to tell you about Parkinson's, actually. So this is some recent results where I've been collaborating with the team of Marie-Laure and Brian Lau and Ken Karashi, where basically, the same principle is sitting here, which is the fact that let's try to resolve as much as possible this variation and use this as sort of predictive machinery to, for example, understand why different patients respond differently to the treatments. So the cool thing is that their team, so they're neurosurgeons, they're specialized in deep brain stimulation, and they've been trying to resolve this for a very long time, the fact that they can get deep brain stimulation to work really well for specific types of symptoms like tremors and slowness of movement. But with respect to one specific type of symptom, which is freezing of gait, which is the inability to initiate movement, which can result in falls and people can get hurt, 1 out of 3 patients on average doesn't improve their symptoms or even worsens. And so they had collected this data set where they bring the patients to the lab, they track their motion, and they make a simple task where the patients move forward, turn around, and move backwards. And they have this longitudinal data set starting from 6 months before the surgery, slightly before the surgery, and then 6 months after surgery under different treatment procedures. And so what we did is that we basically looked at the structure to the differences between individual patients in how they are moving, and identified some interesting biomarkers that are very predictive of the freezing of gait scores post-operatively. So what I'm showing you here color code doesn't matter, but it's the freezing of gait score preoperatively. So if it's high, that means bad. If it's small, that means they're doing okay. So these patients were already selected based on the fact that all of them were big freezers. And they were also selected based on, their responsiveness to levodopa treatment and stuff like that. But so we found that basically coordination patterns between the trunk, the pelvis, and the lower limbs is a very good predictor of how they will respond to the treatment 6 months after the surgery. And we're currently working on ways to implement this into the screening procedure for further surgeries. Yeah, and so basically, the point here is to say that a detailed quantification of behavioral variation, can also be powerful in the clinical context. All right, so I'll just quickly recap, I guess. So I hope I've convinced you that, taking a detailed multiscale approach to the quantification of behavior is powerful and can teach us a lot about underlying control mechanisms. As I told you briefly, we're now working on, in the zebrafish, trying to understand also the neural processes underlying the variation that we see. Again, this is also working towards personalized therapies in Parkinson's disease. What I'm most excited about is also thinking about the evolutionary aspects of this variation. Why is it there? Where does it come from? And so this is work that I'm actively hiring for. So if you know any interested grad students who want to do some behavioral work, please reach out. There's going to be some cool field work to be done in the Indian subcontinent to collect different populations of zebrafish coming from different environments to try to understand basically how evolving in environments with different levels of unpredictability impacts the emergence of behavioral variation over development. So if any of you knows anyone on this, please reach out. And with that, I'd just like to, thank my mentors and collaborators, particularly Claire's, who have been collaborating for a long time, as well Gautam and Leo, and funding sources, and I'd be happy to take any questions. Thank you.